Thanks, Jim. Yeah, I'm sorry, I'm not a believer in title slides, but it has something to do with the biodiversity and the dilution effect as it affects both West Nile virus and Lyme disease. So that's more or less a paraphrasing of the title. And it suggests that I'm going to be talking about biodiversity quite a bit this afternoon, which has an increasingly high profile among both scientists and the public. If you ask members of the public what they have in mind when they think about biodiversity, this slide represents typically what they have in mind. Often they're interested in mammals, birds, sometimes herps, the so-called charismatic megafauna. But when an ecologist thinks about biodiversity, typically what they have in mind is herbaceous plants in a place like Cedar Creek. Or sometimes, if you ask them what they are thinking about with respect to biodiversity, it might include something like a microbial community inside a series of microcosms. Similarly, if you ask members of the public why biodiversity is important, often what they will respond is that biodiversity contributes certain natural products, things that can influence our quality of life and our health. So, for instance, biodiversity is important because things like this poison dart frog have pharmacologically active compounds in their skin. If you ask ecologists why biodiversity is important, typically what they're thinking of is rates of nutrient cycling, rates of primary production, or sometimes secondary production. So what I would argue is that there's quite a strong disconnect between, or a mismatch between, the scientific and non-scientific communities in terms of both the focal organisms of concern and the focal functions of concern. So for instance, the scientific community, the ecological community, as I've just said, tends to focus on herbaceous plants, microbes, and protists, largely because of their experimental tractability, the ability to run well-replicated, carefully controlled experimental manipulations. Whereas the non-scientific community is really interested in very different kinds of organisms. And similarly, the focal functions in the ecological community, as I've mentioned, are things like primary production, nutrient cycling, and the like, whereas largely the non-scientific community is interested in what I call quality of life outcomes, natural products, food, medicines, and the like. So what I'm going to describe is a research program in which we try to bridge this gap by asking this question: whether vertebrate biodiversity, so focusing on the taxa that the public are largely interested in, protect human health. And I'm going to address most of my comments to our research program that directs this question toward the Lyme disease system, but toward the end I will address more recent research on the West Nile virus system. So why would one expect biodiversity should be important in these zoonotic diseases? Well, one might expect this to be the case based on first principles, simply because there are potentially many species that play essential roles in any zoonotic disease. So for any zoonosis, there are at least 3 species that must be involved, and often many more. There has to be a pathogen, there has to be the human victim, and then there has to be at least one wildlife reservoir species as well. But in actual fact, there are often at least several wildlife reservoir species. There's often a vector species involved, or more than one vector species. And then, of course, the neglected component are the myriad other species that interact with that reservoir, the vector, and the pathogen in these natural systems. So before describing to you in some detail how different components, diversity at different components of these systems influence Lyme disease risk, I have to give you a very brief sort of epidemiological background, or I'd like to, into Lyme disease. So it's a little washed out here, but these are 2 patients that are proudly displaying their characteristic, the early symptom, the characteristic bullseye rash, or erythema migrans rash, which is diagnostic of Lyme disease. So other early symptoms of Lyme include fever, chills, muscle aches, lethargy, joint aches, flu-like, generalized flu-like symptoms, but accompanied typically by this bullseye rash. So early Lyme disease, if it's detected quite quickly and treated with an oral dose of antibiotics, is relatively easily cured. But when Lyme disease goes undetected and untreated in its early stages, it can develop into a far more serious late-stage disease that influences the heart, causing carditis, can cause various neurological syndromes, it can cause rheumatoid arthritis, and the like. So Lyme disease is quite a serious health issue in the United States, as well as in Europe and other parts of the world. Lyme disease was first discovered, it's probably an ancient disease, but it was first discovered about 30 years ago, and then in 1982 it became a legally notifiable disease through the Centers for Disease Control and Prevention, which meant that states and counties were required to report cases of Lyme disease. And since that time, the number of reported cases in the United States has grown explosively. Now, some of this increase in the number of cases is clearly a function of increased recognition of the disease by both the public and the healthcare community. But some of that, an unknown proportion of that growth, is due to an actual increase in the number of cases of Lyme, such that by the early 21st century, there were between about 18,000 and 25,000 cases reported annually in the U.S., which makes Lyme disease by far the most common vector-borne disease in the U.S., and there's almost certainly very significant underreporting. So Lyme disease is unevenly distributed across the U.S. There are three main foci, one on the West Coast, another in the upper Midwest, and another one on the eastern seaboard from about Maine to Maryland, or creeping now down into Virginia. Now, this is an interesting observation that has not escaped the notice of politicians, and there are some who think that this is going to become a campaign issue during the presidential campaign that's coming up, and that in fact there's some suggestion that Lyme disease was used as a punishment of liberals after the 2004 election. So we're going to keep careful tabs on just how far Lyme disease spreads after 2008. So Lyme disease is caused by this spirochete bacterium, Borrelia burgdorferi, and that is transmitted to you and me and to other wildlife hosts by the bite of a black-legged tick, formerly called the deer tick, formerly and incorrectly called the deer tick. And this shows an up-close and personal view of a nymphal black-legged tick. And this shows them a little closer to life size. This shows the three post-egg life stages in the life cycle of the tick. So this is a larva, that's a nymph, and that's an adult with a couple of flecks of dirt for scale. Each of these life stages takes a single blood meal from a vertebrate host, lasts for a few days, before then dropping off that host after being engorged, and then molting into the next stage. In the case of larva, molting into the nymph molts into the adult, and the adult reproduces and dies. So there are a couple of key features of the life cycle of this tick that are very important to understanding the ecology of Lyme disease. And those are that the larval ticks, that smallest one up at the top there, larval ticks hatch from eggs free of infection with Lyme disease spirochetes. And that's because there is essentially no transovarial transmission of the pathogen from mother tick into her eggs and therefore into her offspring. So even though we probably get bitten, I suppose you folks do, at least folks working at Duke Forest and elsewhere, I certainly get bitten by many larval ticks, and they don't pose a danger in terms of infectious disease. They're just an annoyance. These larval ticks, though, are very generalized in their choice of hosts, and they will readily feed from dozens of different vertebrate host species. If they happen to feed on an infected host, they may acquire an infection with the Lyme disease spirochete, in which case they will retain that infection through the rest of their lives, and they're capable of transmitting it when they bite a host as a nymph or an adult. So nymphs, it turns out, are the main culprit in terms of transmitting the vast majority of Lyme disease cases to people, because nymphs might be infected if they fed on an infected host as a larva and got infected. They're active in the middle of summer. June and July is the activity peak, and that's when humans typically have their activity peaks outside, or at least in my neck of the woods, maybe here where you're in air-conditioned places in midsummer. And they're also extremely small and virtually impossible to detect when they're crawling on your skin or even embedded in your skin and sucking your blood. So adults might also be infected, but they tend to be much larger, easier to detect and remove, and they tend to be active in the mid to late fall when human activity outdoors is considerably less. So because of this tick life cycle, there are two main sort of entomological or ecological risk factors in the epidemiology of Lyme disease, which I describe in the next two slides. So this is the first one. This is nymphal infection prevalence. And that's simply the proportion of nymphal ticks that are infected with Borrelia burgdorferi, indicated by the red ones on this slide. So what I'm presenting here are two different scenarios. On the left, we have 3 of these 12 nymphs that are infected for a NIP value of 0.25. And on the right, 6 of the 12 are infected for a NIP value of 0.5. And what this metric tells you essentially is your probability of getting Lyme disease given a tick bite. Okay? The other related but somewhat different metric is the population density of infected nymphs. And again, here are 2 scenarios. I'm holding nymphal infection prevalence constant at 0.33 in both cases. And on the left, we have 4 infected nymphs per unit area. On the right, there are 16 infected nymphs per unit area. This metric reflects your probability of encountering a disease-bearing tick given entry into tick habitat. So what I'm going to do is describe the impact of changes in biodiversity first on nymphal infection prevalence and then on this metric, the density of infected nymphs. And now I get to introduce you to my favorite charismatic microfauna, I guess, in this case, the white-footed mouse, Peromyscus leucopus. This species plays an extremely important and underappreciated role in the ecology of Lyme disease, and it does so because of certain characteristics. And those characteristics are as follows. The first one is that it's what I call a highly permissive host. So this otherwise very cute mouse is disgustingly infested with larval ticks all over its ears. And what I mean by a permissive host is that larval ticks that feed on white-footed mice have a very high probability of surviving that larval blood meal and molting into a nymph, as opposed to other wildlife hosts which tend to groom off ticks or have an immune response to the tick mouthparts and otherwise decrease the probability that the tick will survive. The second characteristic is that white-footed mice are the most competent reservoir for Lyme bacteria. And these are data from my lab from a couple years ago showing the percent of larvae that become infected when they have fed from one of these different field-caught mammalian or avian hosts. And what it shows is that about 92% of larval ticks that feed on a wild-caught white-footed mouse acquire an infection, molt into an infected nymph, dangerous to us. And that reservoir competence at 92 or so percent is considerably higher than any of these other host species. Chipmunks and shrews have a moderately high reservoir competence, and all these other species that we've tested have very low reservoir competence. And the third characteristic is that these mice have an extremely large geographic range, essentially covering the eastern 2/3 of the United States. They get into southern Canada and northern Mexico as well. In many of the habitats that they occupy, they are the most abundant vertebrate on the forest floor. So they can be highly abundant, and they're highly resilient. And what I mean by this is that they occupy virtually every terrestrial habitat you can imagine, from old-growth, pristine forests to degraded, trashy woodlots to my basement and kitchen, in fact. And in fact, there's strong evidence that that I'll show you a little later, that they can thrive in more degraded habitats, more so than they do in more pristine habitats as well. So, these characteristics of white-footed mice have given rise to what Felicia Kiesing and I have called the dilution effect model, whereby high diversity protects humans against exposure to Lyme disease. And this dilution effect model is shown sort of in cartoon form in the next couple of slides. So bear with me here. So assume that this is a newly hatched cohort of larval ticks. So these are uninfected, and they sit there on the forest floor waiting for something warm and furry or feathered to walk by. If that cohort of larval ticks happens to encounter a low diversity community, which tend to be characterized by abundant white-footed mice, then they have a high probability of encountering a mouse and molting the next year into an infected nymph. And so what we expect is that when diversity of vertebrates is low, we expect many of the ticks to encounter white-footed mice, and the probability, the proportion of nymphs to be infected also to be high. On the other hand, if that cohort of larval ticks happens to encounter a community that has many other hosts in it, all of which are poor reservoirs in white-footed mice, then we expect the probability of encountering a mouse to be reduced, probability of encountering some non-mouse to be increased, and therefore the nymphal infection prevalence the next year when those nymphs become active to be considerably lower. So if this dilution effect works, then this would constitute a somewhat different, distinct, tangible benefit of biodiversity. And it behooves us to figure out whether the dilution effect operates in nature and how strong is it. So we've been setting about to do that over the past several years. And in order to do so in a rigorous way, I think, we need to determine empirically the role that's played by each host species in this community in influencing total population-wide nymphal infection prevalence. If we can do that, we can then use computer modeling or experimentation to ask how NIP then changes as the diversity in the host community changes. So we've set out to gather empirical information on the role that's played by these different host communities and use a model to account for all the different hosts in these systems. So the nymphal infection prevalence at the population level is simply the number of larvae that become infected summed across all hosts, divided by the number of larvae that fed summed across all hosts. The number of larvae that fed across all hosts is simply the number of mice times the number of larvae on an average mouse, plus the number of foxes times the number of larvae on the average fox, etc., summed over the entire host community. The number of larvae that become infected from all hosts is simply the number of larvae that fed on each host multiplied by that host's reservoir competence, or the proportion that become infected. So essentially, in order to parameterize a model of each host species' contribution to NIP, we need data on three parameters. We need to know how many larvae feed on each host, an average member of each host. We need data on the host reservoir competence, and we need data on the population density of that host as well. So we have spent considerable effort getting the data, and this effort was led by a former postdoc, Kathleen Lejudas. So what we did was to go out into the field and aggressively find and capture members of every species we could possibly catch that occupies the forest floor. And we do this in the summertime when larval ticks are active feeding. And we then retrieve those hosts and bring them back to the lab and hold them in the lab for 72 hours, which is about the time it takes for a larval tick to feed to repletion. We suspend the cages that hold these animals. These are wire mesh cages. We suspend them over water, which allows us to collect the ticks that drop off. We then, and those ticks that drop off into the water constitute one of our parameter values that we call body burden, or B. We allow the ticks to molt, and then we subject them to a test via either indirect immunofluorescence or PCR to determine what proportion of those ticks are actually infected with Borrelia burgdorferi, and that gives us our reservoir competence parameter C. And then either using direct measurement in the field, or in some cases where our estimates were rather poor, we use literature values matched for habitat type and geographic area to estimate population density of each of these host species. And so now what I'm going to do is run through some of these data, but I'm going to go fairly quickly because I think the details don't matter terribly much. But because these data were so hard fought, I do want to show you that we actually do have them in hand. So for white-footed mice, we find that the average tick burden is about 28 larvae per mouse. As I said, reservoir competence is about 92%. And population density varies quite substantially from year to year, but in the year we parameterized the model, it was 25 per hectare. So moving on to chipmunks, and again, I'm not going to go through many of the details. We have a slightly higher tick burden, quite a bit lower reservoir confidence, and about half the population density that mice have. Now, we also parameterized the data for this series of 4 medium to large size mammals, but there are a couple of things that I want to note from this slide. One thing is that deer were the only host we didn't go out and live trap and hold in wire mesh cages over water. We would have loved to be able to do that, but the way we got these data was by collaborating with a local farmer who had a wildlife nuisance permit that allowed him to shoot deer in August, which is when we need these data rather than the typical fall hunting season. So as the deer was falling to the ground, he would get on his cell phone to Kathleen, and she would go pick up the carcass, and we would collect the data otherwise identically. The other thing that I want to note is that this sample size for skunks is fairly low, but I'm actually very proud of that sample. Okay, and then we have short-tailed shrews and a series of 5 species of ground-nesting, ground-dwelling birds that we aggregate for purposes of the model because the sample size for each species was relatively small. And then lastly, we have 3 species of tree squirrels, red, gray, and flying. And then Sorex shrews, which we now have identified as Sorex cinereus. We thought there might be multiple species, but there's only one. So when we put all these taxa together and ask the model, what should the nymphal infection prevalence value be? And we parameterize the model for 2001. Pardon, in this intact forest community, this is what the model predicts we should see as far as nymphal infection prevalence. When we went out in 2002 and sampled the tick population by collecting thousands of ticks and determining the proportion that were infected, we in fact found that the nymphal infection prevalence was within a percentage point of what we predicted. So what this suggests to me is that the structure of the model is sensible and suitable for this kind of exercise, that our parameter values for the individual species are probably pretty accurate, and that we're probably not missing a very important host species out in these communities. We know we didn't catch them all. There are species we were unable to sample, but I would assert that whatever species we missed are probably not that terribly important. So now, given the fit between the model and the data, we can undergo the exercise of disassembling this community on the computer and removing species or groups of species in approximately the order in which we think they disappear under habitat fragmentation in the northeastern U.S. and ask what the consequences will be for nymphal infection prevalence. And that's what we've done now. So if we lose the most sensitive species, Sorex, and these tree squirrels, we find about a 10% increase in infection prevalence. If we lose the other shrew species and the birds, we find a dramatic jump in infection prevalence. That group of medium and large mammals, their loss is actually of less consequence. And then finally, if we lose chipmunks so that we have a community that consists only of mice, our nymphal infection prevalence goes up to the value for mice, which is 92%. So what this tells us is that as you disassemble these communities from an intact one into the most species poor one that you can get, you essentially double the risk of human exposure to Lyme disease, at least by this metric. We've also spent some time more recently asking whether the model, as parameterized in 2001, accurately reflects nymphal infection prevalence both before and after that, since we have measures during that time between 1995 and 2005. And we actually find that there is a significant correlation over a 10-year period between what the model predicts nymphal infection prevalence should be and what we actually find NIP is at our IES sites. Thanks. And there's one sort of, and this correlation regression line is drawn through all of these points. There was one anomalous point, but I don't think we think we know why this occurred, but I don't think I'll wave my arms around that right now. So what I've argued so far is that nymphal infection prevalence is reduced with increasing host diversity. But what about this other metric of Lyme disease risk, this density of infected nymphs? Now, density of infected nymphs is simply the product of the total density of nymphs times the proportion of those nymphs that are infected. And it's possible that as you add host species to an increasingly diverse community, you could boost tick density simply by providing more feeding opportunities for the tick population. So under that circumstance, if you increase the density of nymphs while you're decreasing nymphal infection prevalence, which I think we can argue safely that you would, the net effect on the density of infected nymphs is difficult to predict. So the key questions in asking this is whether in more diverse communities, the species that you're adding simply add feeding opportunities for ticks. So should we use some sort of additive model where different host species add to the feeding opportunities? Or is it the case that added species change the distribution of ticks on hosts without increasing the total number of feeding opportunities for the tick population? So this would be more of a compensatory model. So it's possible that high host diversity could actually reduce nymph density if added species either reduce the abundance of white-footed mouse, the most permissive host, or if they reduce tick burdens on white-footed mice. Okay, so we have actually set out to address these questions, and given how much research has been done on white-footed mouse population dynamics and regulation, I'm a little embarrassed to say that we have next to no information on the impact of either predators or competitors on population dynamics of mice. We know a lot more about bottom-up resource forcing of mouse populations. So we don't have a ready answer in the literature on whether species added to more diverse communities actually regulate populations of mice. So we set out to address this question by establishing 40 sites in what we call the tri-state area, in a focal county in each of New York, Connecticut, and New Jersey, well within the Lyme disease endemic area, to ask this question about whether added species reduce the abundance of white-footed mice. And what we find is that there is a significant negative correlation between the species richness of predators across these sites and the density of white-footed mice, such that when no predators occur, on average there's substantially higher population density of white-footed mice than when there is a rich assemblage of predators. The question about whether added species reduce tick burdens on white-footed mice is perhaps even more challenging, and what we've done, this is work done by a postdoc in my lab, Jesse Brunner, is take advantage of our long-term sampling of both abundance and tick burdens on white-footed mice and eastern chipmunks in our IES forest plots that we've been running for about 14 years now. And what we find is that, in fact, there are a lot of factors that influence larval tick burdens on white-footed mice. So, for instance, the larval tick burden on white-footed mice varies tremendously among years. I don't know if you can see that in the back. We know that males have more ticks on them than females. We know that for juveniles, subadults, and adults, the number of ticks on the animal increases with body mass of the animal. But perhaps most strikingly is that the main factor that reduced tick burdens on white-footed mice very dramatically was increasing abundance of chipmunks, such that as you go from close to 0 to about 80 chipmunks per plot, you reduce the average larval tick burden on an individual mouse by 20 ticks. So our tentative conclusions based on these data are that, in fact, non-mouse hosts can and do reduce the average abundance of mice, and the non-mouse hosts, in the case of the chipmunk data set, can strongly reduce the average tick burdens on mice. And these data would tend to support a compensatory model rather than an additive one. So we can put this all together by asking about the impact of forest fragmentation on Lyme disease risk as measured by both nymphal infection prevalence and the density of infected nymphs. So we've conducted this study in Dutchess County under the expectation that forest fragmentation should change diversity in the host community. And if it does, then we should see a response in these risk factors as fragmentation varies or as fragment size decreases. So our landscape in Dutchess County, some of these colors are rather washed out. It doesn't matter. We have a highly fragmented landscape that has a strong variability in the size of forest patches, from very large continuous forests to very tiny little patches. And we've established a number of different sites in the past couple of years to ask first the question of whether we in fact see a species area curve. Do you see a decline in vertebrate diversity with decreasing fragment size? And in fact we do. It's not as strong as many species area curves are, but we do see a significant decline in total species richness. These are for mammals and ground-dwelling birds. These are basically, this is the show for ticks, as fragment size decreases. So as a result of these observations, it gives rise to a model of what we should expect to see with respect to Lyme disease risk as a function of fragment size.
What's your sort of unit fragment?
Hectare.
Good.
So in small forest fragments, we expect, and I've just shown you some evidence to support the notion that there is a loss of vertebrate diversity. The loss of vertebrate diversity, we think, results in poor regulation and therefore of mice and therefore high mouse populations. That should mean the combination between low diversity and high mouse populations means that ticks should generally feed on the most competent host, leading to high infection prevalence. Because of mice being the most permissive host, we also expect that ticks will be more likely to survive, leading to high tick density. And the combination of high infection prevalence and tick density should lead to high Lyme disease risk. On the other hand, in large forest fragments, we expect that vertebrate diversity should be relatively high. That should serve a regulatory function, modulating population size of mice. We expect that ticks should generally tend to feed on these non-mouse hosts, all of which are less competent reservoirs than the mice are, and that should lead to low infection prevalence. At the same time, with smaller mouse populations supporting ticks, we expect tick populations to have lower survival probabilities, leading to lower tick density, and the consequence should be low Lyme disease risk in larger fragments. So we tested this a few years ago by looking at 14 forest fragments that in which we held constant the landscape context, which is difficult to do because of the heterogeneity of the Dutchess County landscapes. These were all fragments in suburban matrices. And we also held the vegetation type constant as well. And what we found, this work led by Brian Allen, is that the total density of nymphs, irrespective of their infection status, declined significantly with increasing forest fragment size. And it looks as though there was an inflection point somewhere between 1 and 2 hectares or so. In looking at nymphal infection prevalence, we found, in this case, a linear decline, again statistically significant, where nymphal infection prevalence was quite a bit lower in larger fragments than in smaller ones. And in fact, these values for these small forest fragments between 0.6 and 0.8 are the highest published values of nymphal infection prevalence that I'm aware of. And they suggest to me that there was little besides white-footed mice on which to feed for the ticks in those fragments. And when you multiply those two together, you get this graph, which indicates that in fragments that are less than about 2 hectares, the risk of human exposure to Lyme disease, as measured by the density of infected nymphs, was elevated by, on average, about 4-fold. Although there are exceptions, on average, there was a highly elevated and substantially elevated risk of exposure to Lyme disease. So I think our data from the density of infected nymphs support a dilution effect as operating in the Lyme disease system. But you may have noticed that the dilution effect, as I've described it, consists of a couple of distinctly different mechanisms. And just throwing this slide back up again, I want to point out that there are independent mechanisms that are together operating, we think, to reduce Lyme disease risk under circumstances of high diversity. there's this notion that high diversity can regulate the most competent reservoir and the most permissive host. That's one distinct mechanism. Another is that high diversity can actually deflect tick meals away from white-footed mice and onto less competent reservoirs. And then lastly, there's the notion that high diversity can ultimately regulate the vector population as well. So these are distinct modes or mechanisms of dilution, and I think that we need to recognize the dilution effect is not one unitary phenomenon, but actually has distinct mechanisms that can operate independently. So one of our current research directions is, in fact, deconstructing the dilution effect, both theoretically and, if we can get some money, by conducting large-scale manipulations of vertebrate communities, so that we can actually ask which of these mechanisms operates most strongly in this system, which is most responsible for the effect that we see, the protective effect of high diversity. The second of our current research directions is assessing the generality of the dilution effect. Is this something that's unique to Lyme disease, or does it operate in other zoonotic disease systems? And we've been asking this question of late in the West Nile virus mosquito bird system, and that's what I'm going to talk about next. So as some of you are probably aware, West Nile virus is an old world endemic virus that was introduced to New York City in 1999, and it's shown this explosive spread across the continental United States, reaching the West Coast in about 5 years. And this slide shows you the counties in which they had their peak incidence of human cases of West Nile virus over the past several years. So we've been asking the question, so West Nile virus in North America is vectored generally by Culex mosquitoes. There are a couple of mosquitoes, Culex pipiens in the east and Culex tarsalis in the west. The most competent reservoirs appear to be this group of rather common birds: blue jays, grackles, house finch, American crow, American robin, house sparrow, which to me are nothing more than feathered white-footed mice. I mean, I'm sorry, but now a complete list of competent and incompetent reservoirs for West Nile remains to be compiled. So these are somewhat preliminary data, but all these species have been demonstrated as being competent reservoirs. So one might envision a similar role for high diversity in the avian community as we see in the vertebrate host community in Lyme disease, such that if low diversity communities become dominated by a few highly competent reservoirs, then we expect encounter rates between mosquitoes and their main natural hosts the reservoir hosts to be high, and the result should be a high infection prevalence in the mosquito population, and consequently a high number of cases in humans in those local areas. On the other hand, if high diversity communities contain a greater proportion of incompetent reservoirs, they could deflect blood meals away from the most competent reservoirs and result in a lower infection prevalence of mosquitoes with West Nile virus, and therefore lower incidence in the human population. So again, led by Brian Allen, we've asked the question of whether there is a dilution effect. This is done very phenomenologically. Is there a dilution effect in West Nile virus? So we're asking the question, is human incidence of West Nile virus lower in U.S. counties that have a higher bird diversity? And bird diversity we're estimating with the Shannon index. Our database consists of 308, 813, and 30 counties that peaked respectively in these 3 years. And we ran essentially a nationwide regression of West Nile virus incidence. This is per capita incidence in humans, data that are published by the CDC, against bird diversity for that county, data published online in the Breeding Birds Survey by the U.S. Fish and Wildlife Service. So in essence, what we found was very strong support for a protective role of diversity. In all 3 years of the study, we found a very strong and highly significant protective role of bird diversity, such that when bird diversity was very high, human incidence of West Nile virus in those counties was quite a bit lower, and vice versa. And more generally, what we found by using likelihood statistics and model comparisons was, as I just said, the human per capita incidence of West Nile was negatively correlated with avian diversity in that county, and that occurred in all years. Interestingly, human incidence of West Nile was negatively correlated with human population density in all years as well. So the most densely populated counties had low incidence per capita of West Nile virus. But those two factors, despite the possibility that they may co-vary, the models picked them out as both being important and being independently important. So models with both avian diversity and human density had the most support from the data. So evidence for the dilution effect is now being sought and in fact being found, although the strength of the evidence varies quite a bit, in a number of different disease systems. Some of these are vector-borne. Some, like hantavirus, pulmonary syndrome, Bartonellosis, are directly transmitted. Some of these are viral, some are bacterial, some are protozoal. But it looks like it's the case that this protective role of biodiversity may transcend just vector-borne diseases, and it may transcend just bacterial and viral diseases as well, indicating that, in fact, the public's concern for this charismatic megafauna might, in fact, translate into quality of life outcomes for us as translated by the community ecology and the kinds of diversity metrics that ecologists are capable of measuring and putting together. So with that, I will end just by showing the acknowledgements rather than reading through them. There are a number of different people, students, postdocs, collaborators, and on this side, many field assistants, and then, of course, all the funding agencies that made this very expensive research possible. And with that, I'll end and answer questions at this time.